Good afternoon, everyone. So now I think you can hear me clearly, I hope. So we're going to do a bit of a single cell analysis. It was supposed to be today's practical course, and we don't have 2 days. So clearly we're going just to touch the surface of the single cell analysis with our fingertips. And the talk is going to be organized in 2 parts. We're going to see the basics in the first part and a bit of the more advanced, analysis in the second part. So where are we in the single cell analysis process? So we did the sample prep, we did the library prep and the sequencing. And now we are at the analysis step. And in the single cell landscape, there's a lot of things you can analyze actually. And I'm going to focus on RNA transcriptomic single cell and also a bit of protein analysis with the cell surface proteins. And we see also in the second part of the talk how we can integrate different samples coming from single cell RNA-seq data. How we can do the single cell analysis? So we have many options. We can use programming languages, and essentially R and Python are used. Or if you wish, you can also use graphical user interfaces like Schnapps that has been developed by Bernd in the lab, or Cell Ranger Loop.
Sebastien, sorry, yeah. Am I the only one who sees still the first slide?
Maybe there's a problem with the same problem that...
no, that's okay.
So now you're seeing this one, right?
Yes.
Okay. Let me see.
Can you put it a little bit larger? Can you zoom in a little bit?
If I do that, we won't see the title.
Oh, okay. Okay, fine.
And let me... I can try maybe with a... No, that's the only thing I can do for now. I just don't know how to go to the next slide. Oh. Okay, I do that. Okay, I do that like that. Okay. Okay, so we have different options, either by coding directly or using a graphical user interface. Whichever way you're choosing, there's a standard workflow that should be followed, and that's the one we're going to see in this basics introduction. Here are the main steps of this workflow. So first, the quality control, and then the normalization step. After that, we have the feature selection, the dimensionality reduction, and in the end, we will see the clustering. Very often, we will have to, at the end of this step, probably to go back to the first step and redo all the steps. We'll see in which case we should do that. Okay, so let's start with the quality control step. The aim of this step is to select the living cells and eliminate debris or ambient RNA and dying cells. How do we do that? Usually, the quality control is performed based on 3 variables, which are the number of genes detected by cells, the number of UMI, and also the fraction of counts coming from the mitochondrial genes. So for this talk, I wanted to keep it a bit practical, so I used a data set that was available on the 10x data, and that was supposed to correspond to the one that we should have gotten for the course. So that's 10x technology, and it's PBMC coming from human sample. And I'm going to use to illustrate the different steps of the analysis using the well-known SERAT toolkit, but bear in mind that this can be done with other toolkit, the one that you prefer. So the quality control. So there's 3 variables that are important to do that. When we create a Seurat object, we have a table that is created for us that is very important because we have the information for all the cells. You have the identification of the cells here. The first column is the project name that we put here. We don't really need that for the moment. But these two columns are very important because we have the N count RNA column, which correspond to the number of UMI, total UMI detected in the cells. And the N feature RNA is the total number of gene detected in the cells. So among the three variables we need to do the QC, right away when we create a Seurat object, we have already two of them. For the mitochondrial gene expression, it's not very difficult to have this information. We just have to extract the mitochondrial genes in our dataset. So there's a simple grepx function we can use, or you can use an external list of genes. The idea is to have these mitochondrial genes. And there's a very handy function that when you just put your object and the genes you want to calculate the proportion of expression for, do it directly. And by doing just this little bit of code, it directly add, as you can see here in the metadata, a new column corresponding to the percentage of mitochondrial genes expressed in all your cells. So now we have the three variables we need to do a bit of QC. What we can do is to look at the distribution of these variables in our data. And I mean, you can choose something else than the violin plot. You can use a histogram, for instance. But here is what it looks like on a violin plot. So you see, well, no, actually, you don't really see clearly because it's a little bit overlaid, everything. But it's very easy to extract this metadata table and do your own graph. And usually that's what I do. And because I like colors, what I like to do also, it's categorize the mitochondrial gene expression in different classes that are color coded. Like that, I can clearly identify the cells that can be problematic. For instance, the cells expressing more than 20% of their genes as coming from the mitochondrial genes are in red, so you can clearly see them on this graph plotting the mitochondrial gene expression. And when you plot these cells on the other graph with the UMI or the number of genes detected, you can see that these cells tend to go to the lower values of these variables. Another, so that's some can be problematic, these cells. Other graph that can be helpful to find boundaries in these variables is the a simple scatter plot of the number of genes per number of UMI. And then here you can see that you have like a nice compact cells here, and then here it's more like diffuse. So how we can use this graph to try to find boundaries and limits for the number of UMI that we're going to use as minimum or maximum, and same question for the genes. Are we going to put the boundary for the maximum number of UMI here, or here, or here at 10,000? Okay, so we can use a metric for that, which is the median absolute deviation. And basically, what you use this threshold, and this red dotted lines here delimit the cells that are within the 3-time UMI MAD. Okay, this one are larger than 3 times the UMI MAD, and this one are 3 times lower. And you can do the same for the number of genes. And like that, you can select, you see in this rectangle, cells that are in a approximate good range for the number of UMI that you're going to select and the number of genes. So when we have that, we can now subset the SORAT object. So when we use the median absolute deviation boundaries, we end up with 4,030 cells. And when we look at the mitochondrial gene expression in those selected cells, we can still see that we have more than 400 cells with expressing a high number of mitochondrial genes. So we can decide to put a maximum threshold for the mitochondrial gene expressed and use this threshold to discard all the cells having this too much mitochondrial genes expressed. And by doing so, we hopefully we discard all the bad quality cells, and we did not discard important cells. After doing the quality control, we can do the normalization. The aim would be to make the cells comparable with each other's. How we can do that? There are several methods, simple to very complex. And this is an example of a simple normalization that we can do. It's a normalization in the data by the UMI count. So say we have these cells expressing this amount of UMI for gene 1, this amount of UMI for gene 2, etc., etc. When we sum up the total number of UMI, we end up with 6,000. The first step of the normalization would be to divide this value by the total number of UMI. Okay. Then the second step would be to multiply this by a fixed number. So here I chose 1,000. It's just to make the values more readable for us. And in the end, we apply a log transformations that will have the tendency to mitigate the variance mean ratio. So to make the data to stabilize the variance in a way. The other normalization method that is more complex use regularized negative binomial regression. And I'm not going too much into details because it's more complicated than that. But you can see here the results of the different normalizations. So you have on the left-hand side the data when it's normalized by a log normalization using the UMI count, and here using the negative binomial regression. And you can see what you should see here is that in this graph here, the variance is more and in a better way stabilized. So when the mean increase, the variance is still very low, whereas here it's very much more diffuse. Okay. So after the normalization step, we have the feature selection. Why do we need to select the features? So by features, I mean the genes, because many of the genes in the genome will not be informative for a given dataset. And many will mostly have zero counts. That's related to what we call the sparsity of the data. And even after filtering out the zero counts genes after the QC steps, we still end up with, as you can see here on the graph, we still have something like 50,000 genes. Okay. So among these genes, we would like to keep the most informative one and get rid of the ones that are basically noise. So how can we do that? Well, the previous step, the normalization step, is very crucial here because what we did is to stabilize the variance, and now all the genes that are still above a certain threshold of variance will be considered as highly variable, and we are going to keep a certain amount of these highly variable genes to continue the downstream analysis, which will allow to minimize the computational burden on the downstream analysis, also reduce the noise in the data, and help for visualizing the data as well. After normalization, the data, we can scale the data. So the normalization makes the cells comparable with each other. Scaling the data makes the genes comparable with each other. And that's also the step where we can also correct for potential confounding effects. So I say here, see part 2, I was aiming to talk about that during the second part of the talk, but I will not have the time, so maybe for the questions or else. Scaling the data can be done very easily by calculating the Z-score. So for those who don't know what the Z-score is, it's very easily obtained. So here in the table, it's transposed from the previous one. We have the genes in columns and the cells as rows. And say for the gene X, we have the different values of this expression in the different cells. This gives a mean of roughly 150 and 60, sorry, and a standard deviation on 190. So the first step of the Z-score is subtracting the mean. We obtain a centered value, and then we divide by the standard deviation, and we have a centered and reduced value here. What you have to note here is that the overall mean of the genes is 0, and the overall standard deviation is 1. So what we do here is that at the end of this step, all the genes had eventually the same weight. And for some authors, they consider that this may not be really consistent with the biological mechanisms and prefer to skip that step. Also, the way the data is scaled, also using the SCT transform, so the negative binomial regression, is a little bit more complicated than that, and it uses the Pearson's residuals, and it's supposed to be a bit better than just calculating the Z-score. So now we reach the step of dimensionality reduction, and we are going to see what we call dimension in the single cell data and why we need to reduce these dimensions. There's two aims at this step. The first one is to summarize the data somehow, and the second step would be to make it able to visualize the data. There's several steps in this dimension reduction step, and for the summarization, you can use either the principal component analysis or the diffusion map. I will show you an example of the PCA. And for the visualization, you can use either the TSNE that Michel Pusset showed you yesterday in his talk, or the uniform manifold approximation projections, what we call the UMAP, and that's what I'm going to show you now. So this kind of topic of dimension reduction is well known, and not only in single cell RNA-seq analysis. So we use actually dedicated algorithms that were previously existing before the single cell area. So what do we call the dimensions here? The dimensions really are the genes. So here, what you can see on this upper left panel is the matrix of expression of the genes of 5 first genes in 6 first cells of the scaled data. Okay, so at the beginning we have 5,000 cells. So lots of cells and a lot of genes. We have 15,000 genes. Okay. So now, if we want to analyze that, if we have data with only 2 genes, we can do a simple scatter plot, and we can see what difference it can make for the different cells. But we have 15,000 genes. So we already see a step in the dimensionality reduction, which is the feature selection. So we go from 15,000 genes to 2,000 genes, but it's still a lot to analyze. So we have a first round of dimension reduction with a PCA, and we calculate roughly about 50 PCs. So you see here, it's the same cells, but instead of having the gene expression, you have the PCs. And the PCs are linear combination of the genes that are selected, the 2,000 genes, okay? And we can use now these PCs to plot and to project the cells on this reduced space. And you can see here with the two first principal components that we can already see some clusters emerging. So we have like a big cluster here, a small cluster here, and like a big cluster here. So these two parts of the graph being separated by the first PC, the one that explains the most of the variance. So we're going already towards the kind of clustering and kind of nice visualization of the cells, but it's not enough. So we're going to now use a second round of dimension reduction with a nonlinear reduction and the UMAP. And this is the results of projecting the cells on the final coordinates, UMAP coordinates. And you can see that now the cells are tightly connected together, and they form a clearly separated group of cells. We won't call them clusters for the moment. So here you can see on the left-hand side the difference you can have between the PCA and the UMAP, how we can go from one to another. So in this reduced space of the PC here, I just plotted the two first PCs, but I could have plotted more than that. What we're going to do is that in this reduced space, we're going to choose the most significant PCs again, and we are going to use these PCs to for the second round of dimension reduction through the UMAP algorithm. To help you to find how many PCs you should take for this second round of dimension reduction, you can use the elbow plot. So the elbow is where the curve is changing changing. So it should be here roughly around the 10th value. But because the authors claims that with the SCT transform normalization, you can use more PCs than what the elbow plot can suggest. I used the first 20 PCs just to see how it affect the analysis. So just to summarize, so we have here the gene space with just the scaled value of all the genes. Not all the genes, only the most highly variable genes. We do on these selected genes a first round of dimension reduction through a linear PC, and we obtain, we no longer have genes, but we have PCs, linear combination of genes. Among these PCs we calculated, we select some of them that we think are informative, and we put them through the second round of dimension reduction. And here you end up with a table containing only two columns, which are the UMAP coordinates, so axis X and axis Y. And you can project your cells on this axis. So now that we have our cells projected on a nice 2D space, what we would like to do is to find the different clusters, meaning grouping together cells that are close at the transcriptional level. How we can do that? Usually the clusters are obtained by grouping cells based on the similarity of their gene expression. And it's whichever algorithm that you're choosing, there's always two steps. The first is to build a graph-based to build a graph based, on the distance that you can measure between the cells. I took the example of Euclidean distances, but you can choose another way of measuring the distance between the cells. And it's also the number of chosen pieces that is going to be used in these steps. It's not necessarily at the gene level that we're going to do that, but already at the first step of summarization. And in this graph, you apply ad hoc algorithms to find area of densely populated regions. So very often we use community detection methods like the Louvain algorithm, for instance. So you can now attribute for each cell cluster identification, and you can project this cluster ID on the UMAP space, and you obtain this kind of well-known graph with the different colors corresponding to the different clusters. And now the fun begins, because now that we have the clusters, we need to characterize them, meaning associate a cell identity to a cluster. The way we usually do that, it's using dedicated methods to find genes differentially expressed between the clusters. That's one way, that's the unbiased way, let's say. And there's a more biased way if you already know a little bit what your cells are supposed to express. You can use node markers to identify specific cell types. So I'm going to show you both. So when we apply the algorithm to find differentially genes expressed between clusters, we can plot them on a UMAP. And the greener the cells are, I mean the cells of the heat map are, and the more the genes are expressed, and the bluer the less they are expressed. So what we're looking for is like this well-established box of yellow color, and that's not all the genes that are found differentially expressed between all the clusters. The top 10 genes. But already you can see and spot some of them. You see, for instance, IL7R, for instance, granzyme K. So maybe the cells expressing strongly this set of genes here, cluster number 2. Well, maybe this cluster corresponds to T cells, maybe. So that's one way to do and scan this heat map to see if we can identify markers that help us to characterize the clusters. The other way would be to go the other way around. So you already know some markers, and you project their expression on the data. There's several ways to do that. I'm going to show you 2 ways. I could have used the heat map, but I already showed you that, so it's not necessary. You can use for instance, the violin plot again. So here I have a list of marker genes and the cell types they are associated with. And for instance, here for the naive CD4 positive cells, you can see that we have IL7R and CCR7 that are strongly expressed in cluster 0, 7, 9, and 10. 10 for this one, and so on, and strongly in cluster for this one. We can see, for instance, that these genes that is supposed to be specific to B cells is strongly expressed only in cluster 7 and 9. So maybe these clusters corresponds to different subtypes of B cells. And same for CD8A, strongly expressed in cluster 4. So that help us to already have an idea of the different cluster we have in their identity. Apart from the violin plot, we can also project the gene expression directly in the UMAP, and So as you can see, this gene that is supposed to be specific to the B cells is strongly expressed in this tiny cloud of cells here, that if we look at the cluster identity correspond to the cluster number 7 and cluster 9. So in agreement with what we saw in the violin plot. So sometimes it's better to see the expression using this type of graph. Sometimes it's better using the violin plot. So the best way is to use both to have a clear and a nice view of where some key genes are expressed and have an easier way to identify your different clusters. Let's go back to QC now that we have nice clusters. It's also important when you have your UMAP coordinates and you have your clusters established, identified, at least as an ID, not necessarily a cell type, but as an ID, to see if they don't correspond to cells that can be dubious. For instance, cells with high expression of mitochondrial genes expression, or high number of UMI. And what you should check here is the fact that the cells should be together because they share the same biological state, not because they share the fact that they express a lot of mitochondrial genes. It can be both. It can be that a specific cell type express also a lot of mitochondrial genes, okay, or have a huge amount of genes detected or UMI count because it's transcriptionally very active, but it shouldn't be the unique factors that should be taken into account to establish these cells as clusters. So here I can see that we have here a cluster that express a lot of mitochondrial genes because we are above 15%. If we pull out on the UMAP the number of UMI, I could have also put the number of genes detected. We can see that this is the regions that corresponds to a low number of UMI. And here we have a region where we have clustered together cells that expressed high number of UMI. So this is an indication that you should go and look at carefully in these clusters and this one to see if there's a clear marker genes to identify them as a specific cell type, and discard the fact that if they group together is just because these cells happen to have a low number of counts and a high number of mitochondrial genes expressed. So that's where we are. I don't know how long I have spoken, but this is the end of the first part. So I'm going to stop. This is where we are now. I'm going to see if there are any questions about that.
There were some questions in the chat.
Mm-hmm. Okay.
So there's one question Bent answered, but maybe we can share with everybody. It was from Freddie. It was, I was wondering why cells with high mitochondrial genes expression are problematic. The first question. Okay.
That's something that was observed. So in the paper I mentioned, they followed dying cells and characterized them, and it was associated with a high number of mitochondrial genes expressed. And when you have a disruption of the membrane, the mitochondrial gene stays inside the mitochondria and tend to be sequenced, whereas the cytoplasmic RNA is diluted and not sequenced. So they trap, the dying cells trap the reads for the mitochondrial genes.
sorry. There was another question. I know there were some answers in the meantime by Bernd in the chat. It's just to share with everybody because it might be of interest for all. The other question, it was if your computing power allows to, wouldn't you want to like to keep all the features to avoid inducing a bias into your analysis?
So you would need a very strong and powerful computer to do that. And I mean, in a biological point of view, when you have a cell type, it's when you compare the amount of genes that is going to be transcribed in these cell types compared to the total number of genes in the genome, it can be a handful of genes, you know. So it's true that when you choose the number of highly variable genes, I don't think it's a good thing to keep all the genes, all the 50,000 genes, because I don't know a given cell type that will express that many genes of the genome. But it's true that you can choose a different threshold in the number of genes that you're going to select as a highly variable. Here I chose 2,000. I could have maybe go up to 3,000 or 4,000. I haven't had the time to test the different threshold, but it's a fine balance between how much added information these genes will give you compared to the noise they will bring. So it's something that needs to be finely tuned during the process. But no, not keeping all the genes. Yeah. And it's as Bernd is saying, it depends also on the biological questions. If you have, say, in your sample cells that are going to be very different, for instance, you have coming from a brain region where you will get neurons and different types of neurons and glial cells with different types of glial cells. So here, choosing really a few genes will be enough just to separate the big cell types. But if you have a more homogeneous population, then yeah, it will be better to take more genes to find subtle variations in the data. It really depends on the degree of homogeneity of your sample. I forgot to tell you that the part where we identify the clusters is something that should be done with the biologist, because we know, I mean, we cannot know all the different cell types first, and we know what we're doing at the bioinformatic part, but not necessarily the biology. So it's where the analyst and the biologist should talk to see if the clusters found make sense or not, and that's a part where the biologist should be involved also. I see that Anne says that the cells can have different metabolic regimes. And some cells will have a lot of mitochondrial genes expressed. So if that is supposed to happen in your data, you should warn the bioinformatician saying, I don't know what threshold you're using to discard the cells based on the mitochondrial genes expression, but you should know that you can expect to have cells with a high expression of mitochondrial genes. Okay. I'm showing the analysis as if I was blind and using threshold that I'm used to use, and yeah, it's adjustable. As I was saying, once you finish this step, you can always go back and say, oh, maybe I should have gone to 25% of mitochondrial genes, and or being more stringent, and then you redo the analysis and so on and so on. So it's not, yeah, it's you have to fix some threshold at the beginning, but bear in mind that it's not set in stone, and that you can always change that and redo the analysis. It's important to do that, and it's for all the steps. And you saw that I mean I was very shallow in all the steps, and you see already all the parameters you can fine tune. So there's many things you can parameter. When combining samples, when you should use batch correction and when you should not, that's the topic of the second talk. So yeah, we will see that during the second talk. It's a very important question. So that's the part.
Sebastien, can I ask you, if you decide to be less stringent, for example, on mitochondria, where in the analysis downstream you can realize if you were not stringent enough and you have to go back and change your threshold?
Well, so as I was saying, so if you can see now my screen again, so this cluster, for instance, where we can see that the number of mitochondrial genes is highly, I mean, it's elevated. You have other cells with high expressing mitochondrial genes, but they are diffuse, in the data. Here they form a cluster. So you should go and check this cluster, the genes that are supposed to be specific to this cluster. And if you find nothing else than mitochondrial genes, then you can say maybe it's just debris, and do an analysis without this cluster, for instance. And I mean that's where the discussion with the biologist will help to continue the analysis. Like I can't find any specific markers for these cells. Can you think about something? I should go and look in the data to be sure there are cells and not dying cells or debris, and we can try to find a solution. If not, we can do an analysis keeping these cells and an analysis without these cells, and see what it gives in the end. Meaning that in the end, what's important is the validation step, which I'm not talking about here, but it's obvious that everything that we can find by analyzing the data needs to be validated at the bench.